So, until now we have been talking about the different aspects of cell when it comes to tissue homeostasis. So, we talked about cell growth, death, differentiation. So, there are just two more concepts when it comes to cells. The first is adhesion and the next one is migration. So, we will talk about cell adhesion today. So, understanding cell adhesion is crucial when it comes to tissue engineering, because you need to know how you can design your scaffolds in a way that you will be able to help cells to adhere. So, you need to identify what type of cell you are going to work with, try to understand how those cells interact with each other and with the matrix and how they adhere to these surfaces. Based on that, you can actually functionalize your material to get desirable adhesion properties. So, when you talk about cell adhesion, there are the aspects which you need to understand are the concept of the surface receptors. So, each type of cell could have different surface receptors which help in cell adhesion. So, these receptors usually have an extracellular domain, which is the one which interacts with the surface or other cells, and you have a transcellular domain and an intracellular domain. So, through which signals and other cascades can actually be triggered. So, you also have cell to cell adhesion, where cells of similar kind or different kind adhere to each other using some of these receptors and form different junctions, or you could have cell to matrix adhesions. So, it is important to know the difference between the two, and they usually are uniquely different. Some types of junctions are more commonly seen in cell to cell interactions, whereas others are more commonly seen in cell to matrix interactions. So, when you are talking about the mechanics of cell adhesion itself, there can be non-specific physical forces such as electrostatic or steric repulsion or van der Waals forces, which could play a role when it comes to interaction of cells. So, when you are talking about electrostatic adhesion, electrostatic forces, the cell cell interaction can basically have a negative effect, because you can have a repulsion, because there would be a negative charge on the cell surface on both the cells. So, which could have repulsive properties when it comes to electrostatic forces. Whereas, when you are talking about cell surface interaction, you can try to use this to your advantage. If you have a negatively charged surface, then you can induce repulsion of the cells, whereas if you have a positively charged surface, you could actually attract the cells towards the surface. So, steric repulsion is basically seen.
Sir, if naturally two cells both have negative charges and repel each other, like how do they state it?
Yeah, it depends on how far away they are and which forces actually have the biggest impact. So, it is not going to be one force which is acting, right. So, you have electrostatic forces, steric repulsion, van der Waals forces, and then you have the specific interactions because of the ligand receptor interactions. So, based on the distance at which they are present and based on the affinity of these like the dissociation constants or the affinity constants of these forces, you would actually have interactions. So, we will talk about that. So, each of them would have their own properties. So, steric repulsion is basically seen when a cell approaches an adhesion site. What happens is water is excluded, therefore, membrane bound proteins become concentrated, which causes a repulsive osmotic force. So, the compression of the membrane bound proteins initiate an additional repulsive force, which is seen as the steric repulsion. So, again all these forces are more active when the distance between the two surfaces which are interacting, it could be both cell surfaces or a matrix surface and a cell surface, is at different levels. At some particular distances you would have electrostatic effect having a bigger role, whereas at some other distance it will be steric repulsion. So, this the last non-specific physical force is van der Waals forces. So, attractive charge interactions between polarizable but uncharged molecules is called as van der Waals interactions, and this is significant when the distance is greater than 20 nanometers and it is less important when it is very close, because other factors actually play a bigger role. So, this graph which is seen here, the graph you observe here, the interaction potential versus separation distance, this graph shows you how the overall effect of all these three are. So, this is not individual effects, this is a cumulative effect of these three non-specific physical forces. So, electrostatic and steric repulsions are seen when the cells are farther away, and you have the receptor ligand binding which can have a significant impact, and van der Waals binding which would have a lesser impact. Okay. So, the specific physical forces unlike the non-specific ones are the receptor ligand adhesions. So, this depends on what surface and what are the ligands present and what cell and what are the receptors present. Okay. So, the strength is much higher when it is compared to other non-physical forces. Again, this is not a chemical bond which is being formed, it is still a physical interaction. However, the binding is very strong, it overcomes all the other repulsive non-specific forces when this happens, because it is much stronger than all the other forces put together. So, the specificity also provides ways to bind specific cells to matrix to the matrix. So, you could have some cells adhere to a surface, whereas other cells might not actually like the surface as much. So, depending on the receptors which are expressed, you can actually tailor your material in a way that the specific cell types are adhered to the surface. So, yeah.
Sir, these interactions are with respect to tissue engineered cells, or like
No, in general, even in nature. So, if you have an extracellular matrix, the interaction is going to still be there, right.
why are we talking about like distance between the cells? Like in natural tissues, like is there distance between two cells?
Yeah, because the cells, it is not that the cells are always present there and they are proliferating, right. So, cells would have to migrate from the stem cell niche and come to a specific site, and at that, so there will be factors which will play a role. So, it would have an impact even in natural conditions, and it will be crucial to understand that to exploit it in tissue engineering applications. Okay. So, this receptor ligand binding actually controls the binding strength and the binding persistence, because there are limited number of receptors and ligands on the surface. So, based on that you can actually regulate how strong it is and where it actually binds and so on. So, when we talk about cell adhesion in general, we usually are talking about this ligand receptor binding. So, this reversible association of protein receptors in the membrane to the complementary ligands which could be present on the surface or on another cell is what we talk about when we talk about cell adhesion. So, this is what you would have studied when you studied cell biology, right. So, I hope you were taught cell adhesion as part of cell biology. So, it is actually an important aspect when it comes to cells. So, that when we talk about that from a biological perspective, this is what we are talking about. So, if you are looking at it from a chemical standpoint, it is again similar to the ligand receptor binding which we looked at when we talked about growth factors earlier, right. So, what would happen is it is the ligand and receptor forming a complex, and this would be obviously a reversible reaction because it is a specific physical force, but it is a reversible process. So, there would be a dissociation constant and an association constant, and if the dissociation constant is very high, then the affinity is lesser. If the dissociation constant is low, the affinity is much better. So, the interaction would be much stronger. Okay. So, this shows you the association and dissociation constants for different ligand receptor complexes. So, you can see that these are classified based on the type of junctions or adhesions which would be done. So, integrins is one aspect, and you would see, so one type. So, integrins you see fibrinogen interacting with alpha 2b beta 3 receptor, and you see that the dissociation constant is really small. Right. So, that shows that there is a significantly strong interaction. There would be some which are not that strong. So, if you were to take selectins where glycam 1 and L-selectin, its dissociation constant is not that small, it is orders of magnitude higher. So, indicating that it is much weaker. So, there are other things which are as close to even covalent bonds like the biotin avidin complex. So, biotin-avidin complex is as strong as a covalent bond. So, the dissociation constant is about 10 power minus 15. Okay. So, understanding these things can actually help, because specific cell types will actually express specific surfaces, sorry, surface receptors, right. So, we would know. So, fibroblast cell surface will have receptors which will bind with RGD, that is known. So, if you can tailor your material to have RGD, then your fibroblasts can be recruited. So, similarly, you would have to figure out what would be the suitable ligand for different cells which you are working with. So, if you have a bone tissue engineering in your mind, you need to figure out what receptor osteoblasts would like and try to modify your surfaces with that. Hopefully, that will help you help in making sure that more bone cells are attached to the surface. Okay. So, as I already mentioned, affinity is represented by the dissociation constant. The dissociation constant is basically numerically equal to the ligand concentration required to achieve 50% receptor binding. So, this is like your Michaelis-Menten constant, right. So, that is the 50% of your reaction rate is achieved at a particular substrate concentration, that is your Km. So, similarly, the numerical value at which you can get 50% receptor binding is the ligand concentration is the dissociation constant. So, this varies between 10 power minus 6 to 10 power minus 12 molars. So, 10 power minus 6 is considered to be low affinity, versus 10 power minus 12 is considered to be much higher affinity. So, to predict the strength of specific cell adhesion by multiple non-covalent bonds, the tensile strength of the receptor ligand bonds has to be estimated. So, basically to see how strong this binding is, there are experiments to do this, there are many studies. So, there I will give you a reference which you can go through, so that discusses some of the experiments that can be done to study the receptor ligand strength, binding strength. So, the strength of the affinity bond relates to the standard free energy, which is delta G naught, and the bond length. So, what is this delta G? What is Gibbs free energy? At least the ones who are studying thermodynamics this semester.
The spontaneity of the process.
That is the outcome. That is how you. So, if your Gibbs free energy is negative, then the reaction will happen spontaneously. That is fine. But what is Gibbs free energy? Your quiz 2 would have gotten over just now, right? I am pretty sure you would have covered Gibbs free energy.
Already.
What is Gibbs free energy? You studied Gibbs free energy, right? Yeah. So, what is it?
So, it's a measure of the feasibility of a thermodynamic reaction or any reaction.
Okay. So, that is one aspect. So, in general, this is a thermodynamic potential. Basically, it is a thermodynamic potential, and it measures the useful or process initiating work obtainable from a thermodynamic system. So, it could be a reaction or any other process. Okay. Like reaction is what we commonly look at when we talk about Gibbs free energy. So, we always try to associate it with that, but it is not necessary that it needs to be for that. So, Gibbs free energy is the capacity of a system to do non-mechanical work, and delta G basically measures this non-mechanical work done on the system. Okay. Anyway, so the standard free energy can actually be calculated from the binding constants. So, you would get delta G naught equals kT ln of Ka by K naught. Okay. So, Ka here is the binding constant, and K naught is the binding constant at a standard condition, usually 1 mole inverse, and K is the Boltzmann's constant, and T is the absolute temperature. So, bond length is assumed to be greater than the size of the individual weak bonds within the binding site and less than the size of the binding site itself. So, it will be somewhere in between. Okay. So, using this you can calculate the free energy, from which you will have an understanding of what would be the strength of the bond affinity. Okay. So, coming back to the biology aspect of this. So, cell junctions are created when we talk about cell adhesions. The binding of cell surface receptors to complementary ligands is the process in which there is association between the cell and external stimuli. External stimuli could be cells, materials, or cells, matrix, or foreign objects. So, our scaffold would be technically a foreign object, right. So, we would have to figure out how this interaction happens. The stability of these cell or cell matrix assembly can be enhanced by formation of these cell junctions, right. So, instead of just having physical attachments, non-specific physical forces, if you can have these specific junctions which are formed, then you are going to have much tighter and much stronger interactions. So, there are different classes of these cell junctions. So, the first type is called as a tight junction. You also have anchoring junctions and communicating junctions. So, we will quickly go through what these are. So, a tight junction is basically something where closely associated areas of two cells whose membranes are joined together form a virtually impermeable barrier to a fluid. So, this is a very tight interconnected junction. So, the transmembrane proteins interact with each other to form a very strong protein complex. So, here both the cells are contributing equally. So, you would have the receptors and ligands interacting from both cells at an equal level. So, this allows molecules to move against concentration gradient. That is the importance of having these tight junctions. It is not just diffusion through which it is going, it can active transport of molecules is possible through this tight junction. Okay. So, permeability of tight junction decreases logarithmically with protein density in the junction. So, because it is like dependent on the concentration of the proteins, it will be first order mechanism. So, you will have exponential increase and logarithmic decrease and so on, right. So, you would have to see that, you would see that as the number of tight junction, number of proteins or the protein density at the junction increases, you are going to have significant reduction in the permeability, but it will become much stronger interaction, thereby helping in crossing the barrier, like helping in transfer from lower concentration to a higher concentration. So, anchoring junctions are the ones where the protein complex is formed where the cells are actually mechanically attached to another cell or matrix. So, this usually consists of two portions. The first one is the intracellular attachment of proteins which connect the cytoskeleton to the membrane, and then there is a transmembrane linker protein which tethers the membranes between the cells. So, the attachment occurs when this transmembrane protein binds to the complementary protein in the adjacent cell or the matrix. So, this basically acts as a mechanical link, like a hook which has been connected, right. So, now you so this is now connected to the cell transmembrane protein, and then there is an intracellular attachment proteins which connects it to the cytoskeleton. So, it is now kind of hooked to the surface. So, these are some of the anchoring junctions. So, as you can see, you are identify, there are some of these shown here. You have the desmosomes, you have adherents, and you have the hemidesmosomes. So, these are the different anchoring junctions which can actually be formed. So, this is the overview of the anchoring junctions. So, if you are talking about adherents, the transmembrane protein which is involved is a cadherin, and the extracellular ligand is also a cadherin. So, the intracellular linkage which is the intracellular protein, accessory protein which is present, would be actin fibers. In case of desmosomes, you would have cadherin interaction. However, the intracellular linkage is intermediary filaments which are present in the cell. Hemidesmosomes are not cadherin dependent, they form interactions based on with the integrins and an ECM protein. And so, what you can identify from that is adherents and desmosomes are usually used in cell-cell interactions, whereas hemidesmosome is the type of anchoring junction which is formed between a cell and a matrix. Okay. So, again intermediate filaments are involved in the intracellular linkage. So, focal contact is basically an integrin ECM protein junction, which is an actin filament acts as the intracellular linkages. So, integrins are and cadherins are usually involved in these types of anchoring junctions. So, adherents are basically adherent junctions are the type of junctions which connect actin filaments to either the matrix or the cell, and the junctions usually are composed of cadherins which are transmembrane proteins that form homodimers in a calcium dependent manner. So, cadherins require calcium to form these to be active, and P120 binds the membrane region of the cadherin, and beta catenin binds the catenin binding region of the cadherin, binds to the catenin binding region of the cadherin, and alpha catenin binds the cadherin indirectly via the beta catenin or to the cytoskeleton or to the placoglobin, which would be the intermediary filaments, and links it to the actin with the cadherin. Okay. So, these are just the structure. So, the mechanism like the understanding of this details of this is probably not very crucial for tissue engineering applications, but what is more important is to understand how these are formed or like what are the receptors and ligands which are involved in these things, which can be tried to apply to tissue engineering applications. So, desmosomes and hemidesmosomes are also anchoring junctions. So, in case of desmosomes, what you see is a connection between the intermediate filaments to the adjacent cells. The intermediate filaments are usually made of fibrous proteins that form much of the structural framework of the cells. So, some of these are, would be keratin or vimentin or desmin. So, these are just fibrous proteins which are present to create these structural framework. So, the connections here again are mediated by cadherins. Okay. So, these two things, the adherents and desmosomes are cadherin dependent anchoring junctions. So, hemidesmosomes are integrin dependent anchoring junctions. So, here what happens is there is a connection of the basal surface of the cells by means of intermediary filaments. So, these interact with the extracellular matrix to form the junctions. Okay. So, hemidesmosomes, these are some of the representations of how these interactions actually happen. Okay. So, these are the receptors. So, if you look at the major receptors, cell adhesion receptors which are exposed, they can be categorized as into 4 major groups: integrins, Ig like CAM, or cadherins or selectins, right. So, these have different structures, and depending on which cell type you are working with, one or more of them can be overexpressed. So, you can design your scaffolds appropriately. So, basically these 4 can again be classified grouped as 2 major things. One is the calcium dependent and the other is the calcium independent receptors. The cell adhesion molecules which come under calcium dependent would be the integrins, cadherins and selectins. Immunoglobulin like receptors or the IgCAM receptors are calcium independent. So, integrins are calcium dependent proteins which are involved in cell matrix adhesion, but some can actually be involved in cell adhesion as well. So, what happens here is there is a heterodimeric membrane protein which consists of non-covalently associated alpha and beta subunits. So, this is the structure of the integrin, and the binding is actually very specific. It binds to collagen and laminin through the alpha 1 beta 1 integrin, and whereas to fibronectin through alpha 5 beta 1 integrin. Okay. So, depending on the 2 subunits, you can actually have different specificity for attachment. So, So, if you are talking about this transmembrane protein, this connects the cell to the external environment, and the extracellular domain basically binds to the ECM protein, and the cytoplasmic domain binds to the cytoskeleton in the cell. Okay. So, this way you actually have proper interaction within the cell with the matrix. RGD is one of the ligands which interacts with integrin. So, many ECM molecules contain RGD domains. So, this is very popular. Fibronectin actually contains a lot of RGD, so does collagen and laminin and even keratins and so on. So, cell binding through this RGD domain is often mediated through integrins, and so people have actually, this was one of the early peptides which were used for improving cell adhesion. So, when you are looking at modifying cells like material surfaces, people were trying to attach RGD domains or incorporate RGD domains as part of this matrix, or try to use molecules which would have these RGD domains, use proteins which would have RGD domains to prepare these scaffolds. So, that way they were trying to improve cell adhesion. So, it can also be used to reduce cell adhesion if you were to use RGD as a soluble RGD in the media. So, if you want to just move it away from the scaffold, but that is also that might be required if you are looking for like some kind of separation using this like cell separation. So, where you say that cells which do not express receptors for RGD ligands should be adhered, whereas the cells which actually express these receptors should be in suspension. So, if you want to do something like that, then you would use RGD in solution, thereby preventing adhesion of these cells. Okay. Cadrins are again calcium dependent binding. So, these
Can you give an example of it, like why that would be required using RGT to prevent?
So, you remember we talked about cell isolation. So, during that time I was saying one of the things which can be done is to selective adhesion to surfaces or selective separation. So, this would be one of those applications.
So, I have read that RGD motif is widely used in bone tissue engineering. How is it actually?
So, basically fibroblasts, all types of fibroblasts actually have like receptors which will interact with RGD. Osteoblasts are can be have similar surfaces. They are more like fibroblasts present in a bone tissue, right. So, because of that reason they actually adhere nicely to RGD. So, this can be seen with chondrocytes. Many of these types of cells have integrins which can bind to RGD. So, that is why people try to use that. And so, these materials these cells are advantageous because they can also secrete their own matrix, like fibroblasts and osteoblasts or chondrocytes, they secrete their own matrix. So, if you adhere them, so as your matrix, as the scaffold you use gets degraded over a period of time, you would have new matrix formed because these cells are depositing their matrix. So, that is why they try to optimize it that way. Okay. Okay. So, if you are talking about cadrins, these are mostly involved in cell binding, and unlike integrins are mainly involved in cell matrix bindings. So, these are transmembrane proteins which consist of about 700 amino acids. They contain 5 extracellular repeat domains which are called as the CAD domains, and 3 calcium binding regions. They contain a membrane spanning region which is the transmembrane region, and then a cytoplasmic region which typically binds to the actin filaments in your cell. The CAD domain consists of histidine alanine valine domain which regulates the binding. Okay. So, just like how RGD is one of the ligands, here the domain itself contains this histidine alanine valine combination domain. Okay. So, there are different classes. You have E cadrins which are the epithelial cadrins, P cadrins which are placental and epidermal cadrins, and you have N cadrins which are seen in nerve cells, lenses, and heart cells. Okay. So, the dominant mechanism, this is the most dominant mechanism for cell-cell interactions. Selectins are again calcium dependent bindings. These are usually expressed in a transient way. These are not very involved in regular cell adhesion process. So, these are typically expressed on the surface of endothelial cells that line the artery walls, and they contain a lectin domain which recognizes the specific oligosaccharides expressed on the surface of neutrophils. So, thereby they can actually attract neutrophils when there is an inflammation, right. So, this is overexpressed only at that point. So, the neutrophils will bind, migrate and eventually transmigrate through the vessel wall to participate in the inflammatory response. So, that is where selectins play a crucial role. The IgCAMs or the immunoglobulin like receptors contain one or more domains that have structural similarity to the immunoglobulin molecules, and these again mediate cell-cell interactions. These are calcium independent binding. There are two major classes, there is NCAMs and ICAMs. NCAM is when involved in neural cell adhesion, whereas ICAMs are involved in other intercellular adhesion molecules. So, NCAMs are present in many cells, however, they are primarily seen in nerve cells. These bind cells by using homophilic interactions of NCAMs. Whereas ICAMs are expressed in activated endothelial sites into the endothelial cells, and these bind integrins on the leukocytes by heterophilic mechanisms. So, they provide very fine control of cell adhesion. So, depending on the structure of the immunoglobulin which is present, they can actually selectively attach to cells and so on. Okay. So, we looked at what the cells do. So, the other side of it is what are the ligands which are present on the matrices, try to understand that to emulate cell adhesion in tissue engineering. So, we had already looked at what ECM is. ECM is made up of different things, proteins, proteoglycans and glycosaminoglycans. So, glycosaminoglycans could be hyaluronan, chondroitin sulfate, dermatan sulfate, heparan sulfate, keratan sulfate and so on. And you could have proteoglycans and proteins such as collagen, fibronectin, laminin, elastin and tensin, vitronectin, thrombospondin and many other things. Okay. So, depending on what is present, each of them would have different ligands which will help in attachment of cells. The glycosaminoglycans are usually high molecular weight polysaccharides which typically form the ABN, it is a polydisaccharide, right. So, your hyaluronic acid is a disaccharide, a polydisaccharide and so on. So, one sugar residue is always an amino sugar, and the second residue is typically a uronic sugar. So, usually an uronic acid such as glucuronic acid and so on. So, these are unbranched, inflexible and highly water soluble, they are very highly hydrophilic, and they are highly sulfated as well, therefore, they have a lot of negative charges. So, this induces migration of positive charges into the matrix, creating osmotic forces that ensure that the tissue is hydrated, and these are the molecules which are used to form hydrogels even at very low concentrations. So, hyaluronic acid is one example for that. It has been shown to help in embryogenesis, tissue repair, and it also facilitates cell migration. So, proteoglycans are proteins that possess glycosaminoglycans linked, okay, using some kind of a linker protein. So, they possess an extremely high sugar content which could be about 95% of their weight, because the glycosaminoglycans are very large polymers which can be attached to these proteins. So, they are very highly heterogeneous structures and take part in many functions. They can act as reservoirs for signaling molecules or mediators for adhesion of cell membranes or size or charge filters for the molecules which are crossing. So, proteins, as I mentioned, collagen is just the most abundant protein in ECM. There are different types of collagen and they are present in different regions. You have laminin. Laminin has non-specific binding. Laminin is one of the proteins which has very high amount of RGD domain. Fibronectin also has RGD domains which help in integrin mediated cell adhesion. So, other things are elastin, tenacin, vitronectin and thrombospondin. All of these are involved in different aspects and many of them have RGD binding sites. So, as you see, RGD is a very ubiquitous ligand seen in many of the matrix components. So, that is why it has been extensively studied. So, if you were to look up strategies to improve cell adhesion, attaching RGD peptide would be one of the most common strategies. Okay. So